All right, let's dive into one of the biggest questions in all of biology. How in the world do our genes get their marching orders? It's a process that defines who we are, right from the very beginning. So get this. Every single cell in your body, whether it's a brain cell or a skin cell, has the exact same DNA, the same genetic blueprint. So why aren't they all identical? How does one cell know to become a neuron, while another knows to become part of your skin? Who's telling them which part of the blueprint to read? I mean, just look at the difference. On one hand, you've got a neuron, the super specialized cell, firing off electrical signals. On the other, a skin cell, built tough to be a protective barrier. They have completely different jobs, and they look totally different. And yet they both started with the exact same instruction manual. This whole puzzle is really the key to understanding how we go from a single cell to, well, a person. Okay, here's a way to think about it that I really like. Imagine your entire genome is a giant master cookbook. It has every recipe the body could possibly need. Now, a neuron, that's like a fancy pastry chef who only ever looks at the dessert section. And a skin cell, that's the baker, totally focused on the bread recipes. The cookbook is the same, but the chefs pick and choose. So the real question becomes, what's pointing these chefs to the right chapter? Well, the first layer of control is actually written right into the DNA itself. It's like a built-in control panel with these little switches that tell the cell's machinery where to start reading a recipe and where to stop. So one of the most important signals is a spot on the DNA called the promoter. It's basically a landing pad right at the start of a gene. And a key part of this landing pad is a little sequence often called the data box. You can think of it as a big flashing neon sign that says, hey, the recipe starts right here. But it's not just a start signal. Genes also have other control regions called enhancers and silencers, which are kind of like volume knobs. Okay, but, a switch doesn't flip itself. There's gotta be something or someone actually interacting with this control panel to turn genes on and off. So what is it? What are the fingers doing the work? We'll meet the transcription factors. These are the real heroes of the story. They're proteins that act like the chef's fingers, physically binding to that DNA control panel, scanning the cookbook and deciding which recipe gets made today. And these transcription factors basically come in two main types. You've got your activators. They're the gas pedal. They bind to the DNA and scream, more of this one, go, go, go. Then you have the repressors. They're the brake pedal. They bind to the DNA and say, whoa, hold on, shut this one down. It's this constant dance, this push and pull between the gas and the brake in every cell that gives the body such incredibly precise control. So let's just walk through it. Imagine a gene is just sitting there, silent. It's off. Then an activator protein comes along and latches onto the gene's control panel. This acts like a magnet, calling over the cell's main copying machine, RNA polymerase. Once that machine is in place, bam, transcription is on, and the cell starts copying that gene's recipe. And this isn't just theory. It's happening in your body right now. Take your thyroid. Thyroid receptors are a type of transcription factor. Now, when there's no thyroid hormone around, these receptors actually act as a brake pedal. They bind to the DNA and recruit other proteins to help them slam the brakes on certain genes. But when the hormone shows up, it's like a key in a lock. It binds to the receptor, changes its shape, and kicks the brake off. The gene is turned on. It's a perfect example of how the whole body's signals control our genes. But the control system is even more clever than that. It's not just about simple on and off switches. There's a whole other level of control that's more like hiding entire chapters of the cookbook or scribbling little notes on the recipes after they've already been copied. You have to appreciate the packaging problem here. If you took the DNA out of a single one of your cells and stretched it end to end, it would be about 6 feet long. And all of that has to be crammed into a nucleus that's microscopically tiny. So the way the cell packs its DNA isn't just for storage, it's another brilliant layer of control. And this brings us into the fascinating field of epigenetics. The prefix epi just means above. So these are changes that sit on top of your DNA sequence. They don't change the recipes in the cookbook at all. Instead, they change how easy it is to read the recipes. It's like folding a chapter in half so the chef can't see it, or putting a tiny chemical lock on a specific page. Let's look at two of the main ways this happens. First, histone modification.
Your DNA is wrapped around these little proteins called histones, kind of like thread on a spool. By adding chemical tags to those spools, the cell can either loosen the DNA, opening up a chapter of the cookbook for reading, or it can wind the DNA super tightly, effectively hiding that chapter away. The other big one is DNA methylation. This is where the cell adds a tiny chemical group, a methyl group, directly onto the DNA itself. This usually acts like a deadbolt, physically blocking anyone from reading that gene.
But okay, the control doesn't even stop there. What about after a recipe has been successfully copied from the DNA? Well, the cell has yet another checkpoint. First, that copied message gets edited in a process called splicing. The original copy has junk parts, and the cell has to snip them out and stitch the good parts together. And get this, it can stitch them together in different ways to make different final recipes from just one gene. Then, even after all that, there's a final layer of quality control, and that's where these little things called microRNAs come in.
So what do? The best analogy is a Post-it note. After the recipe is copied and ready to go, a microRNA can swoop in, stick right onto it, and basically say, ignore this, or even trash this. It stops a perfectly good recipe from ever being made into a protein. It is the absolute final quality check before the dish leaves the kitchen.
So we've seen this unbelievably complex, beautiful system of control, from switches on the DNA, to proteins that flip them, to how the DNA is packed, to edits made after the fact. It's all working in concert. But what happens when the orders get crossed? What are the real-world consequences when this whole system starts to fail? Well, when this control system breaks down, the results can be absolutely devastating. You can have a gene that's supposed to be off get stuck on, or a gene that should be on get silenced. The cell starts reading the wrong recipes from the cookbook, and that leads directly to disease. And this is not theoretical at all. I mean, look at this. When a gene for a transcription factor like HNF1A goes wrong, you get a specific type of diabetes. When a protein that helps package the DNA, like CHD7, is broken, you get a severe developmental disorder. And when the machinery for DNA methylation fails, like with the TET2 gene, it can lead to blood cancers like AML. Each one is a direct result of a breakdown in one of the control systems we just talked about. And with something as complicated as cancer, it's almost never just one mistake. It's an accumulation of errors. A typical tumor can have, somewhere between 30 and 70 mutations in its genes. And a whole lot of those aren't even in the recipes themselves. They're in the control systems that regulate the recipes. That's what leads to the chaos that defines cancer. And that brings us to the final, and maybe most exciting question. For so long, we've been focused on just reading the genetic code. But now we're finally starting to understand the grammar, the rules of how it's controlled. So the big question for the future is, as we get better and better at this, can we learn to correct the typos? Can we design medicines that fix the broken switches and restore the right orders to our genes? That is the incredible frontier of genetic medicine, and it's the next chapter in this amazing story.